Hey guys, Mr. Dumber coming at you again from Camp Quarantine here in the Dunbrook basement. Hope you're all doing well. We miss you guys. And today we're going to try to get a little bit more done in this PowerPoint. We're almost done with this Unit 6, which is chapters 12 and 13 stuff, DNA and RNA. And today's focus is going to be on basically chapter 13.2. So section 13.2 in your textbook, which is translation. In other words, how does DNA go from being just a chemical code into actually building a protein that gives us a trait. That's going to be our focus today. I'll give you a minute to, or actually you can pause the video and make sure you have your materials ready. You're not going to need to do a summary for this PowerPoint because we're going to have a follow-up worksheet that goes with it on how to translate and transcribe DNA. We'll talk about that a little bit later. Also, you might want to have handy your 13.2 reading packet right now because probably you'll get some answers from this PowerPoint as we go through it. So I hope you're all staying safe and staying healthy and staying smart. We miss you guys, whether you're in the Dunbrook family or the Ward family or the Hazernall or Summers family. We all miss you guys, and we hope to see you in the fall. So stay safe and stay smart. Okay. Okay, so we've been through quite a bit of this PowerPoint so far. And we started off with just talking about what DNA was and how they discovered what DNA did, how they discovered it was different from proteins or lipids or carbohydrates, all that good stuff, right? So we've been through all these slides, so many slides. And we've discovered some of the people that helped us understand this chemical and its importance. Remember, this is the very roots of life. This is the code of how all living things build themselves, and then also how they carry out homeostasis throughout their lives, and finally how they pass these traits or the information for traits down to their offspring, or if you're a cell making another cell, just down to the next cell. So we've been through all this stuff. I'm kind of just scrolling through to get you back on track here. Watson and Crick, Rosalind Franklin, all these people discovered many different things, the shape of DNA being a double helix. Chargaff discovered that A always bonds with T and C always bonds with G. And then we started learning also about RNA, the helper molecule for DNA. And I wanted to remind you also of the analogy that we talked about earlier. But before we do that, as we're on this slide here, we also have been through DNA replication, the way that one DNA double helix can easily unzip and copy itself to produce two double helices that are exact copies of each other so that it can be passed down to different cells or different offspring. Remember through meiosis or mitosis, excuse me. So we talked about replication, and if you're in my class, you also had the models that you cut out, how DNA unzips, and the old left side makes a new right side, and the old right side makes a new left side, and now instead of having one double helix, I have two identical double helices, and this would go on all the way down the chromosome until all chromosomes were copied, all 30 billion base pairs in a human being, all 46 chromosomes, that is. Think those as chapters, and you can then pass those 46 chapters to the next cell or to your offspring or whatever. Then we started talking about RNA in this process called transcription. And remember, transcription was how DNA copies its code to RNA to get its message out of the nucleus, because DNA is trapped in the nucleus and it's safe in the nucleus. And we want to get the message out so that we can make the protein. I will remind you of this analogy that we had. Firstly, looking at the top 10 picture, if you remember this. We're going to compare this top 10 picture to a final product that we want to make. We want a top 10 picture of the kids of the graduating class of 2020, the top 10ers. We want them all to assemble in the correct order and take a picture in the library that can be published in the school yearbook, right? And of course, Miss Hager was in charge of getting all these kids down to the library to pose in the correct sequence to get the ultimate product, which is the picture, all right? So here was our analogy for that. Please keep this in mind. Miss Hager had to send pink slips for each child that she wanted from the top 10 list to come down to the library to have the picture taken. And Miss Hager sent pink slips to do that. And she needed office runners or office pagers to do that. So kids that would run and take the call slips or the pink slips to go get the students. And of course, those individual students would be plucked out of class. They would be assembled in the right order, very important, in the right order in the library. They would pose for the picture, and of course, they would get the final product, which is what we wanted all along, is a correct order picture of the top 10 students for the class of 2020. If you look at this analogy, again, it was 1-to-1. DNA was like Miss Hager. She's the boss. She stays in the office. She's the master plan. You got to protect the master plan. You don't want to damage that, or then you have no plan. So that's why we like to make copies instead of using the master plan, which is why we make pink slips. So the DNA gets copied to messenger RNA. Remember, that process was called transcription. To scribe means to write. So it's like DNA is writing its message down on RNA, on messenger RNA. That messenger RNA is then going to hook up with a transfer RNA. This is kind of like an office page that is going to bring a particular student, an amino acid, to build this protein. Remember, proteins are made of amino acids. So student is to top 10 group as amino acid is to protein. It's just a member of the group. The protein is the ultimate thing that the DNA is coding for, trying to build. Typically, proteins are enzymes. Now, they can be other things like hair and muscle and stuff like that, but most of the proteins in our bodies are enzymes. And what enzymes do is they produce chemical reactions, or they assist in chemical reactions, and they allow chemical reactions to take place in our bodies, in all cells, all living things. And they speed up these chemical reactions, and they allow these chemical reactions to take place using less energy. And we learned that in chapter 2 in biochemistry. So you get the chemical reaction, and finally you get whatever trait you're looking for. So keep this analogy in mind. Miss Hager to the top 10 picture, just like DNA ultimately building whatever trait we're looking to build or make, okay? All right, so that was transcription. RNA, we did a quick comparison of RNA and DNA. Of course, RNA is only single-stranded, while DNA is double-stranded. The other main difference was RNA has a sugar called ribonucleic acid. It's also a 5-carbon sugar, so it kind of looks like a pentagon, as we saw in the nucleotides of DNA. It still has a phosphate, and it still has one of the 4 bases, but it does not have a T, C, and G. Instead, it has a U, C, and G. It does not have thymine. It instead has uracil. So this is all review stuff, right? Remember, the sugar in DNA was deoxyribo, and the sugar in RNA is just ribo, or ribose. So differences between RNA and DNA, the 5-carbon sugar is called ribose. We already said it's single-stranded, which is, by the way, how it fits out the little holes in the nuclear envelope, what we call the nuclear pores. DNA is double-stranded, so it can't fit out those little holes or pores in the nuclear envelope or the nuclear membrane, so it's stuck in the nucleus, and that's a good thing. So RNA is normally single-stranded. It contains uracil instead of thymine. That's very important. So U is substituted for T. RNA can leave the nucleus, DNA cannot, okay? RNA molecules, again, think of them as pink slips. This is actually a transfer RNA molecule here, and the way I remember that is it's kind of shaped like a T, all right? But it's still, you still see those bases, A and C and U and G. Remember, no T in RNA. That's going to become important when we do a worksheet on transcription and translation. There are 3 types of RNA. You already saw messenger RNA in the last PowerPoint. This was like the pink slip. Today, you're going to see ribosomal RNA and transfer RNA. Last time we did the PowerPoint, we did a process called transcription. And transcription was simply turning DNA or copying DNA into messenger RNA. So last PowerPoint, we saw this process right here. DNA unzipping and copying its code into messenger RNA. Today, we're going to see how this code actually builds a protein. So we did transcription last time. Today, we're going to do translation. So much like you would translate English to Spanish or Spanish to English, you take one language and kind of turn it into a meaningful meaning, for lack of a better word, in another language. If you've ever taken a Spanish class or a French class or something, you know that the most important thing is to be able to translate, not just to be able to write the words in a different language. You need to know what they mean as well. So transcription rewrites the words. Translation translates the words or gives them meaning. Again, think of a foreign language. So 3 types of RNA. Here we go. As you know, genes are just sections of DNA in the nucleus that are instructions on how to make proteins, mostly enzymes. Messenger RNA enters the nucleus and makes a copy of the DNA. Remember, messenger RNA can come in and out of the nucleus because it's smaller, it's only single-stranded. The proteins are manufactured on an organelle called the ribosome. So this would be out in the cytoplasm, out of the office, if you will. This would be kind of maybe like the library or something in the school building. All right. A ribosome, again, this is the organelle where protein is manufactured or made. A ribosome is made of many proteins along with, here's a new kind of RNA, ribosomal RNA, rRNA. During protein synthesis, in other words, protein synthesis, look at that word, it means making protein. Transfer RNA brings the correct amino acid called for by the messenger RNA to the ribosome so it can snap them together in the right order. Well, that's a lot to chew on. So let's say it this way. Let's say the office page brings the correct student down to the library so that they can be assembled in the correct spot. Again, the office page, transfer RNA, brings the correct student, the amino acid, to build that protein down in the library, which is like the ribosome. And of course, where did the office page get its information? Well, it got it from the pink slip, which ultimately really got its information from Miss Hager, from the DNA. So the tRNA got its message from the messenger RNA, just like the office page gets its information from the pink slip. However, they all got their message from Miss Hager or the DNA. If you can keep that analogy in mind, it's going to make your life a lot easier. So last time we were doing this, we talked about transcription. And again, it was just DNA unzipping and the left side of the DNA making a copy in messenger RNA, again, like making the pink slip so that this can go out to the ribosome and code for or bring the correct children with the office page or the transfer RNA to the library. And don't worry if you're confused, we'll get into it in a little bit more detail in about 5 minutes here. So notice we're only using one side of the DNA. So the DNA has unzipped here. Let me see if I can focus this a little bit better. The DNA is actually unzipped and we're only copying the left side of the DNA with this messenger RNA. So the right side of the DNA really doesn't do much as far as instructions go. It's just useful to have a template so that we can replicate DNA when we need to have cell division or we need to pass our information on to our offspring, okay? So protein synthesis, again, don't be afraid of words. Protein synthesis just means making proteins. It's another really a way to say translation. What happens here? Well, it separates the double helix and uses the single strand of DNA as a template. A template just means kind of a basis to assemble the correct combination of nucleotides. The RNA polymerase, this is just an enzyme that helps the process, knows where to start and where to stop. This is important, where to start and stop on the entire strand of DNA because the base sequences that signal the beginning and end of the gene. These are called start and stop promoters. Just like if you're writing a paper, you start a sentence with a capital letter, that's an indication that that's a new sentence, and you end a sentence with some kind of punctuation, like a period or a question mark or something like that. Well, the language of DNA also has punctuation. It has places where the gene starts and places where that gene stops, okay? All right, let's watch a quick little video. This is review right here. You guys know A to T, C to G, except for when we talk about RNA. Remember, U replaces thymine. Uracil replaces thymine when we're talking about any type of RNA. If I'm talking about DNA, it's fine to have thymine. If I'm talking about any type of RNA, whether it's messenger RNA, ribosomal RNA, or transfer RNA, uracil is used in place of thymine. So instead of A sticking to T, it would be A sticking to U. All right, and of course, just like when you edit your own papers in English class, sometimes DNA and RNA, when the copy is being made onto RNA, some sections will be removed and some sections will be kind of made more perfect, just like editing a paper. And we call these chunks that are removed introns, and we call the chunks that remain exons. Don't worry too much about that, but just think of it again as taking a rough draft and kind of perfecting it, maybe cutting out some baloney, some garbage out of your paper, and getting a more efficient paper that's a better indication of what you've been trying to say. So again, rough draft, working copy, rough draft, final copy. Okay. All right, let's go to our little video here real quick.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
This is a little repetition for you, but it's going to get into something that we're doing today, translation.
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called Rna Polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus.
Now notice here, again, the DNA, this is inside the nucleus. DNA has unzipped. Now we're not replicating here. We're actually making the pink slip. We're making messenger RNA. So this is transcription. We are transcribing the message. Ms. Hager is writing the pink slip here to get it out of the nucleus, get it out of the DNA, or sorry, get it out of the office.
The DNA code determines the order in which the free bases are added to the messenger RNA.
How would you know that this is RNA being made here? Any guesses? Well, because you don't see a T, you see a U instead. Remember, A sticks to U in this case. Can we still have adenine or A in our RNA? Sure, but we cannot have thymine. So anytime we have an A on one side, we're going to have a U, a uracil on the other side if this is my RNA and this is my DNA.
This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA.
This is the editing portion.
The messenger RNA then moves out of the nucleus into the cytoplasm.
Again, notice, because RNA is only single-stranded, it can get out of these little holes in the nuclear envelope, these nuclear envelope pores. This is also called the nuclear membrane, same thing. Double-stranded DNA cannot fit through those pores, so it's locked up in the nucleus. This messenger RNA is now going to head out to that ribosome to build the protein. So this pink slip is now going to head out to pair up with an office page and a student of the top 10.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer Rna molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time.
Now watch this. This is kind of neat. These are our transfer RNAs bringing our students to the right spot based on these pink slips down here. So this is my messenger RNA. These are my transfer RNAs. Notice how everything is read in groups of 3 letters. Very, very important. Groups of 3 letters. These are like genetic words. There's one word there. There's a second word there. These letters down here are from the messenger RNA. Remember, though, that was really determined by what the original DNA was. We call these 3 letters in groups of 3, we call them codons on the messenger RNA. Up here on the transfer RNA, these are called anticodons. And notice they base pair in a very similar fashion. C to G, A to U, because these are both RNAs. This is messenger RNA, a codon. That word's called a codon. This is transfer RNA. That's called an anticodon. So it's like peanut butter goes with jelly, right? And remember, these transfer RNAs are like the office pages bringing students to the library in the right order. Also notice if there would have been a mistake in the DNA, there would have been a mistake in the RNA, which would have made a mistake in the transfer RNA, which would have brought the wrong student to the wrong place. So mutations, as we call those mistakes, transfer down the line. If the mistake is in the beginning, the mistake continues throughout the whole process.
As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, and there's all of our students in this right order to have their picture taken. So mission accomplished on that one. Mission accomplished. Okay, I hope that helps. Now, proteins are made by joining amino acids into long chains called polypeptides. That's what you just saw. Poly means many, peptide is the type of bond in proteins. Don't worry so much about that. So the word polypeptide really just means protein. Okay? Go back and shut this down. Sorry about that. Back to this. So each polypeptide is a combination of any or all of the 20 amino acids that we possess. And we get these amino acids when we eat protein. When we break down protein, we turn it back into its building blocks called amino acids. There are only 20 different kinds of amino acids to choose from. There's only like 20 different students to build these groups. But remember, we can put these in different orders in long chains, and that's how we can get differences, okay? The properties of the proteins are determined by the combination of the amino acids, what order they're in. That also gives the protein their shape. This combination of amino acids is created, if you remember, from the original DNA code. So Miss Hager's master plan, which was just copied to messenger RNA, sent to transfer RNA, and then those kids were plucked out of class, or those amino acids were brought to the ribosome. Same thing, okay? All right, check this out. Now, don't let this scare you. This is called the Codon Wheel. We have actually cracked the genetic code, which is why this is such powerful stuff. We can now read the genetic code. We know what these letters mean. So if you hear anything today, hear this, because this is going to really help you out with the worksheet that we're going to send you guys with this. This is called a codon wheel. And the way this works is around the edge here, you see different amino acids. These are like different students of the top 10, if you will, or really the top 20 in this case. There's 20 different amino acids. You also see things like stop right there. Remember, we said that the genetic code has punctuation, just like our language has punctuation. So a stop codon would mean the gene is done. You're done building that protein. In other words, that was the last kid to bring to the library. Really, that was the last amino acid to bring to that protein. Here's how you read this codon wheel. This is based on messenger RNA. Remember, we said earlier, DNA, and really RNA, is read in groups of 3 letters that we call codons. Those are like genetic words. So every 3 letters gives you an amino acid. Every 3 letters is like a word. All right. So let me show you how to read this thing. It's actually pretty simple. You read from the inside towards the outside, and you go from big letters to small letters. So again, this is all based on messenger RNA, on the pink slip, if you will. If my messenger RNA was, I'll start very simple here, was GGG, as in grass, 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 you would read it this way: G, G, and there's a G right there. That word, that genetic word, that genetic codon, GGG, means go get the amino acid glycine and bring it to this position wherever that protein is being built on the ribosome, okay? Now notice GGA would also spell glycine, GGC would also spell glycine, and GGU would also spell glycine. So there are multiple spellings for the same word. It's kind of like saying quick and fast. Those are different spellings, but they mean the same thing. All right, let's practice another one. If you had CCU, what amino acid would that code for? What student would that go pick up out of class? CUC, I think that's what I said. CUC. Sorry. CUC. UC would give me leucine. How about UAU? Well, UAU, again, starting at the inside. UAU would give me tyrosine. Now notice I could also spell tyrosine UAC. They mean the same thing. Two different spellings for the same word, okay? What about UAA? UAA would be a stop codon. That would mean the protein is done being built. That's like the end of a genetic sentence. One more. AGC. AGC would give me serine. And notice, again, serine's got a couple of different spellings. I could also spell it AGU. But look over here. Here's another serine. I could also spell serine UCU, UCC, UCA, UCG. So there are 4 ways to spell serine over here and 2 more over here. So in the genetic language, there are 6 different ways to spell the word or the amino acid serine. In other words, there are 20 amino acids, but there are 64 different codons. So that must mean that some codons mean the same thing. Very, very, very important concept. This wheel that you see, this codon translation wheel, really this gives the language meaning. And this is really where the power is, right? We've cracked the code. You may see that in a different format like this. And don't let this scare you. This is the exact same thing that we just did. It's just a different way to organize the information. I prefer the wheel because I think it's a lot easier. But if you see one of these charts, the way you read this is the following. This is the first letter, this is the second letter, and this is the third letter. So let's say I had CAC. First letter C. That tells me I'm somewhere in this big box right here. First letter is C. Second letter is A. So I'm somewhere in this column of this box. So now these are my 4 choices. So if I had CAG, I would have, again, somewhere I'm in here. The letter C tells me I'm in this big box right here in this row. The letter A tells me I'm in this column of that row. And the final letter, third letter G, would tell me I'm at the bottom of that column. So right here, if I had CAG, it would be glycine, I believe that is. These are abbreviated. Or glutamic acid or something. I don't know what that abbreviation is, okay? So again, this is the same information as this. I prefer the wheel. I'm sure you do too. It's a lot easier. Less chance to make mistakes as well, okay? So as we already know, RNA contains 4 bases, A, U, G, and C. And this is whether we're talking about transfer RNA, messenger RNA, or ribosomal RNA. The code is written with only those 4 bases. They carry instructions for 20 amino acids. Again, there are 64 different ways to write this 3-letter code, but there are only 20 amino acids. So each 3-letter combination in messenger RNA, each 3-letter word is called a codon. Okay, codons are 3 consecutive nucleotides, such as ACG or CCU or something. That would be known as a codon. It's like a genetic word. All right. Example. How many genetic words do I have in this messenger RNA strand? Well, I have UCG, CAC, GU. Well, really, that's 3 different codons, which makes it 3 different genetic words, which means it codes for 3 different amino acids. And if you took these codes right here and you looked at the chart or at the wheel, you would see that that's what you would translate those to. Remember, that's what we're doing right now. We're doing translation, okay? And as we said earlier, we have punctuation in the genetic code. There are stop codons, which tells you you're done making that protein. It's like the end of your genetic sentence. And there is also a start codon, which would be like a capital letter to tell you to start a new sentence or start building a new protein. And that start codon is AUG. Anytime AUG appears in the first part of the protein, it's a start codon that says, hey, this is where this protein starts being made. All right. That's about pretty good for today. This last thing here, the decoding of the RNA messenger RNA into a polypeptide chain is, of course, called translation. Remember, polypeptide just means protein. So what we're saying here is when we go from pink slip to actually bringing the kids down to the library, that process is known as translation. Okay? And I'll let you read all this. It's really what we just talked about. Okay, so that's basically the process of translation. I'll let you read this slide by yourself, but really we already just talked about all this stuff. And what we just did was we took the DNA's code, we copied it to messenger RNA, and then we copied that really to transfer RNA and brought the correct amino acids into the correct sequence to build the correct protein. Again, if you look back at the analogy, what we just did here today in the PowerPoint is we had Ms. Hager write the pink slip, get the office runners or the office pages, to bring the individual students, the top 10, into the correct order, to pose for the picture, to produce that final product that we wanted to produce, to put that picture in the yearbook. Really, in real life, what we did was we had the DNA copy its message to the messenger RNA, which leaves the nucleus, and it hooks up with a transfer RNA that brings a specific amino acid to a specific place to build a specific protein that has a specific shape that gives us a specific chemical reaction that gives us a specific trait. Okay, that's what we just did. And these last couple slides, again, I'll let you kind of read them on your own, but we already just talked about this stuff. This today was the process of translation, turning a code into a protein and getting a trait from that protein, getting a chemical reaction from that enzyme. Okay, I hope that helps. Next time we're going to tackle what happens when there's mistakes made in this process, and those mistakes, of course, are known as mutations. If we have a mutation in the DNA that will ripple through the entire chain, I'll show you again up here. If we have a mistake in the DNA, it's going to ripple through this entire chain and build us a different trait. And we're going to talk about that next time. Okay, so stay well, everybody. I hope that you're staying safe, and I hope that you're staying up on your work. Most of you guys are, especially in the Dunbrook family. I appreciate that. Nobody likes this situation. It stinks, but we'll see you in the fall. And please keep doing what you need to be doing, and keep each other safe and keep each other sane. All right. Have a great day, everybody.